Hello, so we are doing DNA profiling. This is just a step from the DNA fingerprinting we did a couple weeks ago. So DNA profiling. It is a process that reveals unique patterns of an individual's DNA, kind of like a fingerprint, only in our DNA. The most important discovery in forensics to date is DNA profiling, being able to take our DNA and see what's in it to figure out who's who. So we have this guy right here. Who is this guy? Well, in 1980, American researchers discovered non-coding regions of DNA. And because of this, this guy, Professor Alec Jeffreys, discovered the process of DNA profiling. That's getting those pieces of DNA to identify a person. And in 1987, 3 years after he figured out this process, a first conviction was based on DNA evidence. So, all right, profiling. Let's get a little bit into it. So, chromosomes are large entwined strands of DNA. I know it's a rehash of bio. They can be divided into smaller sections called alleles. And when common alleles are identified between two samples of DNA, a positive match can be found. So an example, the sample of DNA from a crime scene finds several alleles in common with a sample of DNA taken from a suspect, thus a positive match. So what the heck is going on? Well, cells are broken down to release the DNA. This is called DNA extraction. So here's the blood. The cells are broken down into the DNA, and then DNA strands are cut into fragments. So they're cut up even further, and then they're separated using this right here, the electrophoresis. And a pattern of fragments are analyzed. This entire process is known as electrophoresis. So it goes through this, and then you have your different fragments, your patterns. So some of you have seen this 23andMe, Ancestry.com. Interestingly enough, I just did the Ancestry.com. I'm still waiting on my results. But when is it used? Well, it's used in forensic science, family relationships, healthcare. And in this, some of the things you can see on 23andMe and Ancestry.com is they're looking at healthcare. They want to know about diets. What should you do? What should you be wary of? I didn't go into that part. I just wanted to find, my history. I wanted to see where my family came from. And so I did the basic package, but there were a lot of different packages that had to deal with health and fitness. All right, so going a little bit into the forensics part. So you have this. So right here, this would be trying to figure out a puppy's DNA. Because they can do that with dogs too, dogs and cats and all sorts of stuff, especially for like AKC certified, for show dogs and stuff like that, just to make sure that, this is a show dog. They can look into the DNA. So this is the dame or the mom. This is the puppy, and this is the sire. And you can see when we were talking about the alleles that matched up, well, there's matching. There's matching right there. There's some matching there. There's matching there. And then with the sire, well, there's some matching. There's also matching. And you can see right there, that matches up too. All right, so what can be gathered from the DNA profile to the right? What can you gather from this? Well, here's the victim. Here's the evidence. And then you have 3 suspects. Can you match anything up? Well, let's see. We have a line right up here that matches the victim. This looks like some of the victim is matching. I'm looking across. Yep, lots of stuff from the victim could be matching. Here's the evidence. Let's see if anything from the evidence or the victim matches up. So look at this. The victim doesn't match up with anybody right here. Let's look at the evidence. Evidence. Ooh, it looks like maybe suspect 1 matches up. So you have these 2 spots right here. Why am I not looking at the other ones? I could, but a lot of them match up also. So I want to look at these yellow ones. Look at this. Here's another way you could have it. It's used, DNA is used to analyze family relationships in disputed paternity or immigration cases. So in this case, this is a paternity case. You can see who matches up with the mother and the child. So father 1 matches up with the child when you match up with the mother. Father 2, alleged father 2, does not match up. Another way that it could be used is to provide a valuable information for sea and air accidents when victims are beyond recognition. If they get a little bit of DNA, they can then match it up with a family member to see, hey, is this your missing family member? All right, so common concerns and forensics. The quality of the sample. So forensic samples of DNA are rarely pure. I mean, we were looking at all the other evidence types so far, and DNA seems to be the best one to analyze, the best one to find, to match up with people to people and not have a lot of mistakes. So typically bloodstains on clothing or furniture will be contaminated with all sorts of other materials. DNA from fungi, bacteria can be present, and shows up in fingerprints. DNA decays rapidly, so some of the DNA sites attacked by restriction enzymes may be lost or cause them to be to cut in wrong places. Other contaminants may attach to the DNA fragments and weigh them down, and which shifts those bands, the ones that we just saw, into in an unpredictable extent. Mistakes and inaccuracies. Well, human beings collect these and store them, so there can be human error. Accuracy has been challenged because segments rather than complete DNA strands are fingerprinted, so fingerprinting may not be unique, and often tests carried out in private labs may not follow uniform testing standards or quality control. Interpretation. Human interpretation can lead to errors. Banned on electrophoresis gels are sometimes indistinct and can result in interpretation being a matter of opinion. Evidence from criminal investigations. If certain behavioral family relationships or physical characteristics are shown to have genetic roots, a DNA database could offer the opportunity for discrimination. When DNA fingerprinting is used in criminal investigations, should authorities be allowed to keep that DNA on record, or samples of those people not proven guilty, or those who are proven guilty? Should they be allowed to? That's one thing that could come up, that has come up, that has been a debate, because it's found later, in several years later, that the DNA matches that this person gave not knowingly. What organization might want to use this, the DNA, and who's going to cover the cost? So cost effective. Who's going to pay for it? One other thing I wanted to bring up is distribution of alleles frequencies in various VNTR loci can vary among different ethnic groups. So that's another thing to keep in consideration. So you have this right here. I'm not going to play it right now. I will link this in the for the classroom thing. I'll link it. And finally, gel electrophoresis is a complicated and expensive lab. Today you will complete a virtual electrophoresis lab experiment. You're going to be using Adobe Flash. I'm going to leave a link to help you enable Flash. You need to use Google Chrome, especially for those of you on school computers. Once you learn how to perform electrophoresis, you can now learn how to read the results, which we will be doing a little bit later. All right, so that's it for the notes. Make sure you have taken the notes. If you have any questions, hop on Zoom. Remember, my Zooms are always 2 to 3. You're required once a week to show up. That's 5 days to get in once. Besides that, if you have any questions, class tag me, email me, hop on Zoom, ask me questions. Besides that, hope you guys have a great day.